Okay, I think we'll perhaps get started. I've got various things to show you. I'm going to move around cameras, etc. a bit, so please bear with me if it all gets complicated. But I'm going to start off by sharing my screen. I want to thank you all for coming and also for your support throughout this series this year. I've really enjoyed hearing all the talks and I hope you'll enjoy this one. Right, so if I share my screen and go full screen, can you all see my screen okay?
Yeah.
Perfect. Okay, so I think many of you will have seen this picture. We've used it a lot for the COVID support group, and it's been all over the news, etc. And what it is actually a fairly accurate representation of what a coronavirus looks like. In red, you've got these spike proteins, which will be the subject of today's talk. In grey is the phospholipid membrane, which is the same type of membrane that covers all of our cells. And then in orange and yellow are two other types of proteins called the N protein and the E protein. Now, this picture was actually made by two people at the Centers of Disease Control. You can see their names down in the bottom here, Lisa Eckert and Dan Higgins. It's fairly correct. They have slightly downplayed the number of E proteins, and they've given it a sort of sinister lighting because they wanted everyone to be very scared and to stay indoors, etc. That's at least what they say in the interviews I've read with them. So what I want to do today is to try and tell you how the information behind this image was come by. And it's a story that dates back to right at the beginning of the last century. And much of it is rooted in Cambridge, and quite a lot of it is rooted in the MRC Laboratory of Molecular Biology, which is where I'm sitting at the moment and where I work at the moment. I'm not going to talk very much about my own lab's work. That's a story for a different day, but I want to focus really on how we know what proteins and other molecules look like. So perhaps if I move on, the first thing I wanted to do was give you an idea of the scale we're talking about. So what I've done here is shown you a human hair that's about 0.1 of a millimeter wide, or 100 micrometers. And I've blown it up so that it should fill quite a lot of your screen. Now, if you look to scale, one of the cells, for example, that coat your skin, they're about 10 micrometers wide. And this picture here is roughly to scale. I can probably bring a laser pointer on to show you. And then a coronavirus also to scale is this tiny thing down the bottom here. Now, if you blow that coronavirus up and then look at these spike proteins, they are then blown up here. And so this spike protein is what, about 10 nanometers across, whereas the coronavirus is about 200 nanometers across. So you see we're getting smaller and smaller. You can imagine how tiny it would be compared to a human hair. And then what makes up a spike protein are amino acids, and what makes up amino acids are atoms. So on the right here, you see a little tiny corner of this spike protein, and it's made up of this chain of amino acids. And you can see I'm tracing the backbone of the chain, and off the backbone are all these side chains, which you may be able to see are made of different molecules. So this one, for example, has a yellow sulfur in it. This one is a ring of carbon atoms. This is called a phenylalanine. And so proteins are made of these long chains, thousands of amino acids that fold together to produce these proteins that do work. So towards the end of the talk, I'm going to say what the spike protein does. But first, I want to go into how we know what things look like at the sort of atomic level. And it goes back, as I mentioned, to the beginning of the 20th century. And a guy called William Lawrence Bragg, who was born in Adelaide in Australia and came back to England to go to Cambridge at the same time that his father, who was I think a physicist, came back to Leeds to take up a professorship in Leeds. Now, Bragg graduated in 1911 from Trinity and then started a PhD. And in the first year of his PhD, he made this really amazing discovery that transformed our ability to see atoms. And you can see this is the paper he published from that work. And he's listed as W.L. Bragg, BA. So he hadn't even got his PhD by that stage. Now, what happened was that a few years previously, a German called von Laue had put a salt crystal into a beam of X-rays and had seen what was called a diffraction pattern. So that's these spots here that surround the central X-ray beam. Now, what this experiment had told von Laue is that X-rays are made up of waves. It had been the proof of that. And Lawrence Bragg's father, who was also called William, he was William Henry Bragg, he had sent Lawrence the paper, and Lawrence had been sort of walking around Cambridge and suddenly realized that he could figure out the atomic structure based on the pattern spots and how he could do that. And then he and his father together collected images like this one you see here that allowed them to work out the structure. And the famous one, of course, is sodium chloride, or salt, common salt, which is made up of this lattice of sodium and chloride atoms. So let's step back a bit. Diffraction, so X-rays are diffracted by this lattice of atoms. That's actually somewhat similar to the diffraction of light and the diffraction of other waves. And to try and explain that, I'm just going to try and get fancy with my experiments. So first I'm going to stop sharing, I think. And I'm going to change camera. Here we go. Right, okay, you see here. What we've got over here is a laser pointer, just the sort that you would use for a talk if you were giving a talk. And then down here, we've got a slide, and in it we have a metal circle, which I'll show you in a minute, close up what that looks like. And it's just a grid of spacings. Now, when we fire the laser at that grid, the waves of light are going to be diffracted by the grid spacings. So that instead of seeing sort of the grid pattern, what you see is this pattern of spots, just as you would with the salt. And in order to do that, I need to change so that you now see this image, hopefully. This is my phone, which is sitting over here, and it's just pointing against the wall. So if I point the laser just at the wall, you see that's the laser light. If I shine it through the grid spacings, you see this amazing pattern of spots that comes out. And that is caused by the diffraction of light as it sort of interferes with this lattice of grid bars. Right, let me come back to myself, come back to sharing the screen. That's fine. You've seen light being diffracted by a grid, but what does that actually mean? I think, so the grid I just showed you is this one. It basically looks like this. It's this round circle with metal grid bars. What is diffraction looks like in terms of waves? These come from much older experiments that some of you may have come across before. This is called a wave tank. And what you have at the back is a paddle that goes up and down and generates waves. And they're going to come towards this slit in this block. Now, what we're going to do is we're going to look at those waves by panning down and looking at light that comes through this water tank. And it will show us the pattern of waves. So if we pan down, this is just a video I got on YouTube. What we're going to see is a pattern of waves that comes towards this gap and then get diffracted by it, and then the video will stop. Okay, so what this shows is you've got these parallel waves that come out, they go through this gap. Instead of just going through as a thin beam, they actually spread out. And this is the process of diffraction. It's because of the way they're crowding and trying to get through that gap. Now, if you then move to a lattice, I 2 points or multiple points, you start seeing the basis of this formation of spots. So in this case, we're going to do the same thing. We're going to look at some waves and then pause the video. You can see there are the waves and they spread out. And what I hope you can see if I annotate them is that these are the high intensity waves that come out, and they are where the waves reinforce each other and add up to something bigger. And then in between them, you see these sort of very flat areas, and that's where the waves cancel each other out. And so you see nothing. And that then is the basis of diffraction and the spot pattern of spots that you see. Let me just try and move on. Okay, so the same principle is causing this pattern of spots here. What Bragg worked out was that he could use the spacing between these spots to work out the distance between the planes of atoms in his crystal. And so it was that realization that allowed him to work out the structure of this simple molecule. Now, I've shown you a picture on the left of him as a much more elderly man, because after he'd had a very distinguished career, he served in the First World War, he won his Nobel Prize at the age of 25, which is still almost the youngest. There was that lady from Afghanistan who's younger, but the youngest science Nobel Prize ever. He had a distinguished career, and right at the end of his career, he went to London to work for the Royal Institution and head it up. But he really missed his garden. He had, I think, a house on Grange Road or somewhere with a nice garden, missed his garden. So he took a job as a gardener at one of the sort of London houses. And apparently he called himself Willie and said, yes, ma'am, and no, ma'am, and touched his cap and had a lovely time gardening. Until one of the ladies' guests said, my dear, what is Sir Lawrence Bragg doing in your garden? And at that point, he had to resign because it wasn't deemed right to also be a gardener. But I think that's sort of, you know, it's a lovely story about it. What he did before he went to the Royal Institution is he sort of oversaw and helped support a number of people that moved the field from looking at these crystals of you know, small numbers of atoms to much more complicated systems. And I want to tell you about 3 people. The first is J.D. Bernal, who had actually been a PhD student with Lawrence Bragg's father in Leeds. Now, he was the first person to start crystallizing much more complicated organic molecules with many more atoms in them. And then that was taken a step further by his PhD student, Dorothy Crowfoot, later Dorothy Hodgkin. She was the first person to actually crystallize a whole protein, pepsin, and with Bragg worked out that it would diffract and produce diffraction patterns. And then the next PhD student, so that was in 1934, the next PhD student was Max Perutz, who had had to escape from Austria. His family were Jewish, he'd had to escape the Nazis, came here, did his PhD with Bernal. But the problem with Bernal was that he was an ardent communist, and this was not appreciated by the sort of senior people in Cambridge. So they promptly sort of didn't give him a job. And so Bernal had to move to Birkbeck, where he had a very successful career. And Max then came under Lawrence Bragg, where he finished his PhD. Now, Max then went on in subsequent years to work out how to go from these diffraction patterns of protein crystals to actually solving an atomic structure of them. And it turned out that protein crystals, as you'll see in the next few slides, have much more complicated diffraction patterns. But from them, you can work back to the actual structure. Even though there are multiple atoms in this lattice, you can work out that structure by calculating both the intensities of those different spots and also something called their phase. And I'll tell you a bit more about that in a minute. But first, I wanted to let Max tell you a little bit about his project himself and how he was working on hemoglobin and this phase problem. So I'll just play this movie, which again, I got from the internet.
Research students in the lab thought I was crazy to take this on. But then, you see, I reflected that hemoglobin may be 1,000 times more complex than the structures they were working on, but it was also 1,000 times more interesting. Now, you notice two things. The spots are arranged very nicely and regularly, and that merely gives you the distance between the hemoglobin molecules in the crystal. But then you see that the spots have different degrees of blackness. Some are very dark and others are quite faint. And that we call the intensities of the spots. The problem is that measuring the intensities gives you only half the information needed to solve the structure of the crystal. The other half is called the phase. They gave beautiful X-ray diffraction pictures. So those thrilled me enormously, you know, and I would show them to all my friends, say, but look what marvelous X-ray pictures I got from these hemoglobin crystals. And when they asked me what they meant, I would change the subject because I had no idea.
So Max, indeed, at that stage had no idea how to interpret it. But after about 16 years of work, he did manage to figure it out. And it turned out that what you had to do was take your crystal, collect a data set on it, then soak it in a heavy metal compound like mercury. And that changed the pattern of spots sufficiently so that you could work out the position of all the density that gave the protein, shall we say. So basically what he was doing is measuring the intensities of these diffracted waves, but also working out their phase. That is where the peak of the wave is when it hits the detector relative to the atoms that it was diffracted from. Okay, so what Max got out of that was this model that he computed using many of the early computers, the valve computers that were in Cambridge. And he got this model of hemoglobin. Hemoglobin is the protein that carries the oxygen in our blood. And he was incredibly proud of this model. I was a PhD student in the lab when Max was still around. And when I first walked into the LMB, this model would sit in reception. And then if Max gave a talk about his stuff, he would come and get it and bring it in and sort of point at it. And it's actually, I've been given special permission to bring it here and to show you. So I will change my video yet again and stop sharing. Hang on. Sorry, playing with technology. Right, let's have this one. Okay, you should be able to see that. So this is Max's model. I borrowed it from the library at the LMB. Let's see if we can get it into focus. So they didn't have computers. So in order to visualize it, he made it of balsa wood. It's a little fragile, but it's still pretty much intact. What you see here is 4 different chains that make up hemoglobin. And then in red here are the hemoglobin that have an ion in the center that actually attaches the oxygen and carries it. And Max spent his career working out how this works. It's terribly beautiful. As you bind more oxygen, it becomes easier to bind the next one, which helps you pick up and release oxygen in the tissues and the lungs. And I just wanted to show you here because this was the first structure of the protein ever. And it's a great okay. So that to Max, let me just come back to my other camera. So that was the first protein structure that Max solved. If I return to my script, you see that he spent his career working on this protein. By 1984, he was able to get a very highly detailed model with all of the atoms correctly in place. And so this is the same sort of image, but now with the backbone of the protein traced in these ribbons. And you can see the heme groups and the iron in orange here. And so he spent his career working out. Let me just... Sorry, mute a couple people there. So what happened next is, in order to do this, he needed the support, and that support, thanks to Lawrence Bragg, came from the Medical Research Council, and they founded a unit for looking at protein structures that turned into this Medical Research Council for Laboratory for Molecular Biology, which is the institute we're sitting in now. It started off in this hut in the courtyard of the Cavendish. This hut survived until about 7 or 8 years ago, and it's now, I think, a bicycle parking site. Then in the 50s, they moved out to this purpose-built building on the Adam Brooks campus. This was before the hospital was built. The hospital would be here at the end of this image. This is now, you can still see this building. It's been extended, it's surrounded by other buildings, but it's still there. And then in 2013, we as an institute moved to this new building by the railway track, which you can see. And at the moment, if I turn on my laser pointer again, we're sitting down in the basement here of this wing of this building. And so what happened over the next amount of time is that crystallography got better and bigger and more sort of able to solve all sorts of interesting proteins. One of the sort of almost the largest or some of the most exciting things that it was used to solve were structures of the ribosome. And I show here Venki Ramakrishnan, who's also in this institute. And he was my PhD advisor. So in the middle here, you see a crystal structure of the ribosome. You can see it compared with the size of hemoglobin, so it's much, much bigger. Now, the ribosome is the complex in the cell that generates all the other proteins. It reads the genetic code, generates proteins, and is sort of essential for creating all of the machinery that's needed for the cell to survive. It's made of proteins, so here I'm shown in orange and dark blue, but it's also made of lots of RNA, which is shown in light yellow and light blue here, that forms the core of this structure. Okay, so I just wanted to show this because my PhD project indeed was to work on it. We worked on the yellow part here, the small subunit. There was a really exciting project because at the time it was by far the largest thing, it and the large subunit were the largest thing that had ever been crystallized. And again, if I stop sharing, I just wanted to show you what that looks like in 3 dimensions. Can you see my screen okay with this molecule? Probably you can't see me, but
We'll see you, Andrew.
Wave if you saw that molecule. Okay, so what this is the ribosome. You can see the RNA in green and all of the proteins surrounding it. And then as you zoom in on here, I want to show you some electron density.
We can't see Andrew. We can't see it.
Face, we're not seeing your
Seeing
screen.
you, Andrew.
Seeing it. Sorry, let me try and share. Can you see it now?
Yes, thank you.
Yes.
Technology, sorry.
Yes.
Great. Okay, let me turn that off and zoom out. Right, okay, so this is the small subunit of the ribosome. In green, you see the RNA. In different colors, you see the proteins. And this was a project that took 5 of us in the lab working on. I only contributed to this, but we were able to solve this structure during my PhD. But my role in this was to soak the protein, the ribosome, with antibiotics. And one night I processed the data and saw these antibiotics for the first time. And I just wanted to show you what that looked like, because it really was incredibly exciting. So what I had was the blue chicken wire here is the density that you calculate from those diffraction patterns. So this is the representation of the atoms. And that might become clearer if I show you this, sorry, as bonds. Right, so now you can see these are the individual atoms that have been modeled into this density. And as I move it around, you get a 3D idea of it. And what I've done as the experiment was to soak in 3 antibiotics. And you see 2 of them here. They're shown up in green. That's a technique we use to show up things that are missing from the rest of the structure. And then if I zoom in on here, I can then put in that final structure, and you can see that this small drug molecule fits perfectly into this density. Now this molecule is streptomycin. That was the first antibiotic ever to be shown to be active against tuberculosis. It was what changed tuberculosis from being an absolute certain death sentence into something that could be potentially treatable. And about 50 years after that discovery, which was in the 1940s, we saw it for the first time, and that was magical. As with these discoveries, they often happen late at night, and this one, I first saw this density very late at night in the basement of the old building. Right, I'm gonna move from there back to the screen. Can you see my main screen again?
No, not yet.
No.
Stop sharing and share again. Okay. You should, I hope, now speed back to thank you, Ramakrishnan, and the ribosome.
Yes, thanks.
Perfect. Okay, so until about 2010, 2013, crystallography was the best way of looking at proteins. But then electron microscopy became used to look at protein structures. Now, electron microscopy had been around for a while, and the idea is that you can take electrons, which are usually found in atoms surrounding the nucleus. You can strip them away with high voltage, make a beam of electrons, but you can treat this beam similar to you can light, because it can be focused by electromagnetic lenses in a way that X-rays can't. X-rays can't be focused, but electrons can. So what you can do is take an intense electron source, put it through various lenses, goes through the sample, and it generates an image. And that's very similar setup to a light microscope. Now, electron microscopy had been used for years. It was actually instrumental in naming coronaviruses. So coronaviruses were discovered by this, first isolated by this guy called David Tyrrell, who worked at the Common Cold Unit, which was a center in Salisbury, where you would go for a free holiday and they would give you a cold, and you got a paid holiday and you got sick, and then they did some experiments on you. And it closed down in about 1990, but it did all sorts of important work on colds, and it was where coronaviruses were first discovered. So David Tyrrell sent his sample to a lady called June Almeida, who was an electron microscopist. Here's her sitting in front of an older version of this microscope that's behind me. The electrons are coming down here, they're being imaged at the bottom here. And she took these first images of coronaviruses. They were stained with the tungsten stain, a phosphotungsten stain, to make the protein stand out white against this dark background. And what you can see is they look a bit like sunbursts. And so they were named coronaviruses because they had this sort of sun-like appearance. And this sun-like appearance, of course, are those spike proteins that we saw at the beginning. So what became apparent, though, is that you can also use electron microscopy for looking at protein structures. And this, again, has been done for a number of years. What you can do is take your protein in a buffer. You can put it on a grid like the one I had over there that I shone the laser through. And then you can plunge it very quickly into liquid ethane. And that freezes it so quickly that the water forms this vitreous glass-like substance. And then you can put that in the electron beam, collect images, which are these 2D projections of your proteins. And then you can use computers to work back from all those different projections and work out what the original 3D structure should look like. Now, for a long time, people were able to do this, but the sort of resolution they got was always quite low, similar to the hemoglobin model I showed you, where you can sort of see the shape of the proteins, but not any of the detail you wanted. And one of the people who changed that was Richard Henderson, who's also here at the LMB. He, for many years, had worked on proteins by electron microscopy. Here he is holding a model, a low-resolution model of bacteria rhodopsin, which is a little pump that pumps ions across a bacterial membrane. And you can see here, you can see the sort of low-resolution features. But he sat down and did calculations and worked out if you collected your electrons with a much more sensitive detector, then you would be able to solve structures to atomic resolution. And he worked with companies and others to develop this detector. This is the first one. And that is what then revolutionized electron microscopy. So the big machine behind me looks amazing, but actually it's the fairly small camera down in the base of it that revolutionized the method. And so here, for example, is the spike protein. In the middle, you see an example of the raw image you'd collect with these very faint, fuzzy images, these dark bits. Those are the spike proteins. But when you average them all together and average tens of thousands of them together, you can get this amazingly detailed images. So you can see here perhaps the fine details of the protein chain. And into that, you can then build models like this one down here. This is the spike protein that I showed you earlier. It's got 3 protein chains. It's got a whole load of sugar molecules in gray surrounding it. So what can we do? Now we have the structure, we can actually start explaining how it works. And the role of the spike protein is to bind to proteins on your cell surface and allow the virus to get in to you. And in order to tell you about that, I'm going to show you a series of animations. And these were created by Janet Iwasa. Janet was a PhD student in San Francisco when I was a postdoc there, so I know her quite well from that time. And she has a career where she helps explain science by making these animations. So for example, here is an animation how this virus particle moves. You can see the spike proteins were waving around. You can see the other membrane proteins in that embedded in the membrane. Then what we're going to do next is have a slice down through the movie and have a look through the virus and have a look inside. So cutting through. This then is the genetic material of the virus. You can see that it's RNA, so just as in the ribosome, that contains all the genetic code to make new viruses. And it's wrapped up around this other protein called the nucleic acid protein and sort of stuck like this slightly frightening worm-like structure in the center of the virus. And so let me show you next a movie in which I'm going to explain or in which Janet, I think, will explain how these spike proteins allow the virus to get into the cell. I'm just going to stop sharing and reshare and make sure I've got the sound on, which I have, and I'll play this movie.
After inhaling airborne viral particles, these viruses can then travel into the airways where they may encounter cells of the respiratory epithelium in the trachea and lungs. The spike protein on SARS-CoV-2 recognizes a specific protein called ACE2, which is shown here in purple on the cell surface. The spike protein, which is normally very dynamic and able to undergo opening, closing, and bending movements, is locked into an open position after binding to ACE2. Another protein on the cell surface called TMPRSS2 and shown in orange can then cut the spike protein in a specific location. These segments of the spike protein fall away, exposing a portion of the spike protein which was previously hidden. The spike protein is then able to undergo a series of dramatic conformational changes. During the first stage, the spike protein inserts itself into the membrane of the cell. In the second stage, segments of the spike protein zipper back on itself, forcing the membrane of the cell and the viral membrane to fuse. After fusion, the viral RNA is deposited into the host cell, where it will direct the cell to produce more virions.
Right, so that then is an explanation of how we first get the coronavirus. It comes into our lungs, and that is the process by which it gets into our cells and then starts replicating. So that is a wonderful illustration. Scientifically, it summarizes a lot of information. But in the last few minutes of the talk, I wanted to tell you some of the uses of that information in the pandemic and in fighting SARS-CoV-2. And the first example is given by this person called Jason McClellan. Now, he's been studying coronaviruses for a long time, and back to the SARS and MERS epidemics that were a few years ago. And he solved, I think in 2015, 2016, a cryo-EM structure of the spike protein. And what he realized is that when it undergoes that transformation you just saw, it's because these helices here, which you can see folding over, unbend and straighten up. And so what he realized is if he made these mutations in these positions that are in red, he could actually stabilize them in this sort of folded down conformation. And that makes the protein more stable. It's less likely to go into this post-fusion state. And it's therefore much more likely to be recognized by the immune system and your antibodies when it infects you. Now, those two mutations were used to stabilize the protein in a number of different vaccines, particularly Pfizer and Moderna. Interestingly, they weren't used in the AstraZeneca vaccine. And so they're clearly not essential for the vaccines to work, but there's some evidence that they may help in stabilizing the protein and therefore in the production of vaccines. People are working on various other ways of stabilizing the protein based on the structure. They're taking out that cleavage site that the temporous protein cleaves. I actually spent quite a lot of lockdown working in the lab trying to introduce disulfides between cross-links between different parts of the protein in order to try and stabilize them. And a number of other groups have done similar things, and those are also being used to stabilize the structure. So a number of sort of structural-based ways of working on this protein that have been important in helping in that fight for vaccinations. But there's another example I wanted to tell you about, which actually I came across on the BBC only a couple of days ago, and I thought was nice to also include. And to explain about that, I just need to show you that, of course, it's not just the spike protein that's made by this virus. So the virus itself has this long RNA, which is shown here in black, and that encodes a whole load of different proteins. The ones down this end, the ORF3 and the spike protein, are all separate proteins. But on the left here is encoded this incredibly long protein shown in blue that's then chopped by another protein up into all of its separate components. And the protein down here, this heart-shaped one, it's called the main protease. That's the one that chops it up. So if you could inhibit that protease, you would have a really good drug to help you fight against COVID. And that's exactly what has been made. This was something which you may have seen on the BBC. It's called Paxlovid. It was developed by Pfizer. Its name here is PF07321332. And it was developed during the pandemic. It's now being sent out to people who are particularly susceptible to COVID. If they get it, they're sent this drug, and it can help reduce their chance of getting severely ill. And this drug was generated using structural information. Over 80 different crystal structures were solved in its development, and it was developed incredibly quickly. Usually a drug like this would take 8 or 10 years to develop, and it was done in 21 months. And they started off, for example, at the beginning of lockdown on the 13th of March, with this original candidate molecule, which they knew inhibited the protease from a different coronavirus. And the problem with this drug is that it could only be given intravenously. So over the next couple of months, they modified it and used structures and designed bits and made lots and lots of compounds, 800 compounds, in order to come up with this one down the bottom, which is what's in this final drug molecule. And so again, it was really structure and knowing what this protein looked like that enabled them to go so quickly there. I also just wanted to mention very cleverly, I think, this Ritonavir has got no activity against SARS-CoV-2 at all, but they add it to the mix. It's actually an HIV medicine, but what it does is it so occupies the enzymes in the liver that they don't have any spare capacity to break down the active compound. And so it's used basically as a blocker to allow the active compound to stay in your body a bit longer. Okay, so I've told you about crystallography, cryo-EM. They've both been important in the coronavirus outbreak and in many aspects of modern biology. But what's really exciting is that we've undergone, I think, a new revolution in structure in the past actually year during the lockdown. These two people and the team behind them have begun to be able to predict proteins from their just raw sequence. So without having to solve structures, you can very accurately predict proteins. It's not perfect, doesn't work in every case, but it really is remarkable what they can do. Demis was a student at Cambridge. He got his A-levels at the age of 16. He then had to do a gap year because Cambridge told him he was too young to start. So he went to work for a gaming industry, computer game company for a few years, and then came up to Cambridge and then moved on and founded this DeepMind company, which uses artificial intelligence to solve all sorts of problems. And one of the problems that they turned their attention to recently, and that was a team headed by John Jumper, was to try and fold proteins de novo. And I won't go through the details, but here in blue, you see their prediction of one of the COVID proteins, and in green is some experimental density. So you can see they're really very accurate. Now, what this means is that going forward, we may be able to solve all the proteins. Well, we should know all or most of the proteins in us, and that could be incredibly useful for working out which drugs are going to bind in the correct one and have an effect and not bind all of the others. That, of course, will take a lot of work in terms of working out how small molecules bind these compounds, but I think it's a very exciting way forward. Okay, and so with that, I'm going to stop, and that's just letting me turn around, and I'm going to stop sharing and take any questions if you have them, and if I can hear you, and in a bit, perhaps try and crack open this machine behind us. So thank you all for your attention. And are there any questions?
Andrew, I saw a program on BBC Horizon the other day. I think I pointed them out to you. It was quite an interesting one because it followed the chase to develop a vaccine. And there was and this struck me because there was that Australian group who I know the building very well, having come from there. And they introduced a bit of HIV to the building of their mimicking the spike protein. Why would that, and it actually ultimately became their downfall simply because when they were doing testing, like they do an LFT, they were showing HIV proteins were showing up. And so, of course, nobody wanted to take it, of course. So it was killed almost straight away. Why would they use the HIV? Do you know?
No, and I should have watched it. There are definitely links between the HIV proteins on its surface and the spike protein. This idea of cross-linking or of making those two red mutations that I showed actually came originally from HIV. So what it might have been is that they were using the base of that molecule as a sort of support for expressing the spike.
Yeah, they were. It was showing HIV at the point where the spike protein joined the cell.
Yeah, so actually most people were using some trick to support that region. I mean, the work that we were doing, I can't even remember what it was now, but we had some artificial linker at the bottom that was helping hold it together into the three chains. And so probably it was something along those lines.
Yeah, it's a really interesting one, and if people are interested, I'll put up the links, because it was a fabulous following of the research groups that were chasing the vaccine.
Thank you.
How automatic is the analysis of the structure from the crystallography, from the X-rays?
How automatic? In terms of getting the electron density is incredibly automatic now. I mean, often it will flash up whilst you're collecting the data. They've got processing pipelines that will, do that. When I was a student, it was a bit slower. There was software for doing everything. I mean, back in the sort of early days, they were calculating everything by hand and then on computer, and then writing the first software themselves. By the time I was a student, the software was always in place. By the time I set up my lab in 2010, it was drag and drop. You would take your file and sort of drop it into a window and it would show you your structure. And now I think it's even more automated. In terms of building proteins, that is actually one of the great things AlphaFold has done, is allowed you to have very accurate starting models, and then you can sort of sort out the final little bits that are not quite right. So things are becoming quite quick.
Right, thanks.
Hello, is Alan? You're muted still?
Who, me? No, I'm not muted.
Oh, Alan Painter.
I'm muted.
Oh, sorry.
Sorry.
Andrew, do you use NMR technology at all in your analysis?
It is used. I haven't actually used it, I think, ever, but many of my colleagues do. It's particularly useful for flexible and unfolded proteins and dealing with them. It's able to get information about them which no other technique can use. So it has traditionally been used alongside the other methods. I personally haven't used it so much, but it is very useful for these smaller, flexible bits.
Presumably the X-ray technology is more readily available. It's probably cheaper, is it?
It's not. It's partly that. So with X-rays, the problem is actually getting crystals. For some things, it's very easy to grow crystals of proteins. They grow in 15 minutes. Other things can take years to get anything to grow crystals. That's why electron microscopy caused a sudden sort of burgeoning of what we could look at. Suddenly we could look at things which would never have crystallized, it would have taken ages to. With NMR, there was quite a strict upper size limit because of the way the calculations are done, and because the protein, NMR requires the proteins to be tumbling very quickly in a solution. And if it's too big, it just tumbles too slowly and you don't get sufficient signal from the different atoms. They basically get blurred out if it's not tumbling around fast enough. So that's why NMR wasn't used for the same number of sort of proteins that crystallography was typically, but it is used. Dave.
Thank you.
I can't, sorry, you're muted.
First of all, bloody brilliant, loved it. Secondly, did you notice the change in how you had to do your work because of the pandemic? Because you were, I'm going to use the phrase, pootling along, right? And then pandemic hits and it is incredibly important. And did you have to make some decisions on how you did your work based on the situation?
Yes, so you know, some of us directly contributed to the effort to sort of work on the coronaviruses, and some of us tried to keep on going with what we were doing generally, to sort of keep things ticking along. Actually, in my case, my neighbor was getting live viruses in, was doing all this work on the spike protein. He was a sort of proper virologist, structural biologist. And so I volunteered to help him. And he said, no, I've got a PhD student who will do it. I don't need your help. And then, unfortunately, the PhD student had to go home. And so he said, well, actually, could you give me a hand? So I spent quite a lot of the pandemic in the lab doing stuff for that. And then some of my people joined that project, some of the people in my group, but actually a lot of them then were continuing, you know, trying to continue the projects that we did mainly. So I think that's the balance, that there were people working on it, contributing, but some of the basic work continued as well.
So to hog the thing, would you do anything, can you see your future being different because this has happened, that it might happen again? Do you see what I mean?
Oh, I see what you mean. I mean, I have quite a long-running interest in viruses, and so I think we will continue that. I'm interested in how they're moved around the cell. I think one of the things that really struck me was that what was important was the background work. So the most influential person in some ways that I've talked about for the coronavirus was that guy, Jason McClellan, who had been working on it for years at a time when nobody was interested in coronaviruses, right? All they did was cause the common cold. And yet his sort of insights from that work then suddenly became incredibly important to the vaccine developments, etc. So I think one thing that I would say is that for academic science, it needs to carry on doing that stuff, which seems really boring, because you never know when it's going to be super important. I think that's part of the message there. Great.
Andrew, can I just ask about the MRC and the funding? Is that sort of central government funded or sort of medical sort of part of government? Or do you rely on Wellcome Trust and people like that for funding as well, like many other scientists?
Yes, so this institute is funded by, it's now UKRI, and MRC, is Medical Research Council, is part of that. So that's money that comes directly from central government. That funds the institute, a lot of the infrastructure, and some of the people in our labs. Some of us have Wellcome grants and other grants as well, and that typically would produce a lab that's a bit bigger. So there is a certain core structure, and some people run their research programs purely on that government money, and others have a few more people funded from elsewhere.
Thank you.
But one of the amazing things about this place is because it has that central funding, there's a great deal of sharing about it. You know, if you've got a chemical, it doesn't matter if the person next to you borrows it. And so things are really shared around the building. And that's, of course, terribly efficient because, you know, you don't buy the same chemical 10 times. You get it once and then, you know, share with people, etc. And so I think that's why this has been a traditionally very good model here.
And is there pressure on publishing results and things, as there is in many scientific programs within universities?
There's always, I mean, one of the reasons there's always some pressure to publish results is you have students and postdocs who are young people who, you know, want to move on with their careers and want to publish their things. And the other thing is science moves at this amazing, very rapid pace. And it is actually important to put out your information so that other people can build on it. And so that, you know, other people are probably working on aspects of it at the same time. So if you never put anything out, you know, somebody else will do it. And actually that's a waste. You know, you want to contribute your ideas and help keeping the field moving on as much as you can. So I think that's where the pressure is, shall we say.
Thank you.
Cool. Okay, any other questions? Yes, sorry.
Hello. My name's John, and very, very simple question. My understanding is that the old MRC had a cafe bar or a restaurant, and that's where a lot of the chatter took place, and that was very useful. Does the new one have such a facility?
Yes, it does. The L&B canteen is famous, particularly for its cheese scones and its chatter. They did, when they designed this new building, the canteen was a very important part of that. And it's on the top floor, it looks out towards Trumpington, and it's where many people gather. And quite often, if you want to meet somebody to discuss a project, you pop up to the canteen with them. A lot of discussions happen there. It's perhaps not quite the same in the days when the Institute was so much smaller that everybody knew everybody. But it's still a very good place to bump into people and keep those interactions going.
And you're not protective.
Sorry?
You're not protective.
No.
Yeah, you will chatter genuinely to each other.
Exactly, and yes, that's true within this institute. It's one of the great things about it. It's been a great pleasure working there because of that.
Thank you very much.
Right, shall we crack open this electron microscope?
Mm-hmm.
Let's see if I can do this. Right, I'm going to go to my phone if I can find it. Hang on. Has it died? No, it's here. Right, where are you, Andrew? Here we are. Spotlight. Okay, right. You should now see my phone, and you can still hear me. Yes, that's good. Right. So there's the model. This is the electron microscope. So if I press this button, open up. This is, and you can see it's a huge. I mean, you saw that before. It's a huge machine. And also the facility manager very kindly opened this side as well, so we can really take a good look. Right, okay. So what we've got at the top here, which you can see, I guess where I'm pointing to, that's the electron source, 300 kiloelectron volts electron beam. That then comes down through this column that you can see. And what you're seeing here is the various different lenses. So the condenser, the objective, which is here, and then looking down, you get the projection lenses that then magnify the sample. The sample itself, I can try and show you. It's on one of those grids. Here. So this was the sample preparations. Place. Everything, of course, is done under liquid nitrogen. And what you have is you have those grids, and they are then placed into a structure like this. This is the AutoCAD that's where you put the grids, and then you put that into this structure, which we normally put a liquid nitrogen. You would bring that to the machine and place it down. I should say that this machine is not, it's shut down at the moment for repairs, which is why we can do it. And then you push that in there, you push it at the back, and then a robotic arm would come down. You can just see, perhaps through this little window, that's where the robotic arm is. That would come and grab that cassette, take it up, and then push it into the vacuum, which is where the electron beam is going. So, and all of it is kept cold by this dewar you see here. That's all full of liquid nitrogen that's automatically refilled by the large vat of liquid nitrogen that's a back there. And then I hope this is not making you too dizzy, but if we go round the back, you'll see all of the electronics that drive this. There's quite a lot of development done by somebody in the past in terms of the electronics driving it. You can see all of the wires and the cables on the floor. And then if I come here, facility manager also kindly opened up the back of it for us. And this little thing down here is that's it's called through. That's the magic that allows these colors to and then you can actually see that it's at right angles to the main column, and that's because there is a filter that actually bends the electrons when they get to the bottom of the column. It bends it down, it gets rid of the white switch. We want to make okay, so I will come back. And then here you've got, it's all driven, of course, by computer. The screen on the right is off, but you can see the controls that are used for focusing the electron microscope. And then this is the sort of image that you would get of one of the holes with the ice in it that would have the sample in. On the right here is an overview of the whole grid. You can see the spacing of the metal bars, and that has a carbon sample with the protein on it. And you can see the little green dots where someone has selected the area that they want to collect. You can also perhaps see just how sort of broken up it is, even with things that look like very bad samples. Sometimes you can still collect data. Okay, I will stop that.
Yeah.
And I think we should wrap up. Thank you all for coming and listening. That's the last of the Know Your Neighbours for this year, but we can think about doing it again next year, but that really depends on whether you think we should. So if you do, let Alan or I know, and we will try and think about